Okay, so we are now going to cover what are the neuronal mechanisms that are responsible for peripheral vision. And here we are aim to explain the property of the visual system from eyes to V1 and explain the color vision at the periphery. So this is just a recap of the retinal structure going, the lights are going from top and then goes through the layers of the ganglion cells here, and then reaches in the end to cones and rods area, and then these layers are intermediate pre-processing areas, bipolar cells, macroin cells, and then reaches to ganglion cells, and then axons go like this way, and the axons are this yellow thing on the figure. And then if you do the neurophysiological technique, then you can record the neuronal activity from this retinal structure. The important thing is to insert the electrode into the eyes. And here, the eyes doesn't actually have to be inside of the live animals. Eyes can be isolated, but still respond to the lights. So, for example, glass electrode can be penetrated like this and then attached to the ganglion cells in the retina. And then you can record the spike that looks like this vertical line here. It's called a raster plot. And at the same time, you can also potentially measure the same axons that goes from here to here, and then measure the spikes at the optic nerve level here. And then basically this scheme shows that, looking at the same, as long as you're recording from same neuron with the axons identified, then you see the same pattern of the spikes. And the spike is, as you can see, on and off, and then the pattern of the pulse and also temporal and spatial pattern of the pulse is conveying the information about this visual information. So, and what you're learning right now is this very important concept of the receptive field. Okay, and in particular, in the case of the retinal ganglion cells, receptive field is important. So each of the line corresponds to the particular response over 2.5 seconds of the visual stimulation. And then the top is when the particular retinal ganglion cells recorded are shown by the black thing under their so-called receptive field. So here it is, in a sense, the baseline firing rate of the retinal ganglion cells. And then when you present the spot of the light here in the left side of the neuron, and then this neuron actually spikes a lot during the stimulus presentation time here during this on to off period. Okay, and if you convert this spikes into the sound, then it sounds like that's the neurophysiological recording. And then if the optimum size of the stimulus as a white spot exceeds, and then if it stimulates too much, then it doesn't produce as much like kind of response. On the other hand, the optimum location, in the optimum location, if you present the black thing, on the other hand, then this becomes inhibited. It becomes below the baseline. So one would conclude that this particular neurons that we are recording from, has a receptive field that looks like the center area that becomes activated by the positive or sort of the bright spot, and then it's suppressed by the dark spot in the center. On the other hand, the annulus of this center area, because it inhibits compared to this central spot stimulation, this area is called the inhibitory area. So most likely this neurons receptive field looks like this. Center is activated by the white spot and inhibited by black, and the surround is suppressed by the white and then less suppressed by the black. So that's the receptive field of this particular neuron. And then similar thing can be applied onto this analysis of these neurons. Here, the baselines again identify the black spot, and then white spot onset inhibits this neuron. But interestingly, here releasing the white spot into the black makes firing here. Okay. And then white circle, bigger circle, doesn't produce much. It's similar to the spontaneous rate. And then now the black center white annulus pattern excites the neuron a lot at the onset, and then at the offset it becomes slightly inhibited. So two things about this neuron. So this neuron's receptive field is likely to be that center is excited by the black, and then also excited by the white ring here. But this white ring excitation is strongly inhibited by the white center, and then that's consistent with this one. And then also one thing I forgot to mention on this thing is that there are two types of the response in terms of time. Stimulus onset response, which is easy to understand in terms of mental kind of picture here. That's usually called onset response. Okay, and then at the offset of the stimulus here or here, that's called offset response. And typically, retinal ganglion cells have, in the end, have a structure called on-center or off-center. So on-center corresponds to the left and the off-center corresponds to the right, and also a combination of onset response or offset response. So at least there are 2 by 2 kind of response matrix, on center or off center, and then onset or offset response, and they are all found in this neuron. And this onset and offset response is pervasive across all the cortical areas, and we don't usually talk about offset response, but you can assume that many of the neurons have this onset and offset response types. Roughly, I'm not sure whether the exact proportion is computed, but maybe half and half could be assumed to some extent, because nothing counts as a background. So background is onset for something, and onset for some cells could be considered as a background. Let's move on. And then this is another important figure, and I'll explain. So this is the visualization of the retinal ganglion cells dendrites on the visualized on the surface of the retina. And the one type of cells called a mediate cell, that means small, and the parcel cells that are bigger in terms of like a big parcel. And the mediate cells are usually connected with the cones, and the parcel cells are connected with the rods. And the parcel cells are related to the detection of the motion or low contrast stimulus. As you can imagine, some kind of functionality that goes with a bigger receptive field. Basically, the size of the dendrite also corresponds to the extent of the receptive field. Okay, and the mediate cells are small, and so it's correlated with sort of the fine resolution and also color type experience. But exact computational wiring is not known exactly, at least in the human. Okay, and then the important thing is that first, the size of the neurons are indicated by this, scale. So here, roughly, it's a 115 micrometer diameter neuron. And this is much bigger, roughly like 250 micrometer neuron. And then the numbers below here, 1 millimeter, 5 millimeter, or 10 millimeter, these correspond to the distance from the fovea. Okay, so if you remember this, eyes, figure, fovea corresponds to somewhere here where the axons dips here, and then at the 1 millimeter from here, you find relatively small one for both midget type and the parcel type, and then 5 millimeter you get bigger retinal ganglion cells, and also 1 centimeter, 10 millimeter, you find even bigger neurons. Neurons with bigger dendrites and also bigger receptive field. And this is very important for consideration of our discussion. Okay, so what it means in terms of our fovea peripheral vision. So as I said, the central retina, closer to the retina, each of the ganglion cells actually projects to almost one-to-one kind of no transformation is happening from retina to the next stage of the visual processing called the lateral geniculate nucleus. And I will spell it out in later. And then at the level of B1, peripheral primary visual cortex, these the LGN neurons tend to converge onto one neuron. So B1 neuron's receptive field is even bigger than the retina or LGN, and it also has a line-like shape, as I explain next. But importantly for our discussion of fovea versus periphery, the peripheral neurons are already bigger in terms of the receptive size at the level of the retina, and then it converges on the V1 neurons like this way. So V1 at the level of V1, the extent that it can collect the lights of a particular wavelengths, like yellow, can be actually bigger. So when I presented this type of the figure in the demonstration, last week and also at the beginning of this lecture, when red dot is very small but presented at the fovea, it can project to certain clusters of the retina and then excites a particular neuron in the B1. And if these guys prefer or generate color experience of the red, most likely it's not even like B1. After that, B2, B4, and so on. And then current theory says that B4 or B8 is likely to be the color generating center of the cortex. But by that time, when the signal gets there is some kind of convergence of the information, but still, all these areas of the neurons are looking at particular relatively small areas of that vision, and our perception is reflecting on that small part of the visual field. On the other hand, the big chunk of the yellow here projecting onto retina of this wide side, that can effectively stimulate these neurons which prefers yellow light, let's say, and then that projects to LGN, and that converge onto one V1 neurons. And you can imagine that this thing actually goes on to B4, B8, and this generates yellow sensation. And there is nothing magical about that. So what it means is that as long as the stimulus is presented in a large extent or in proportion to the scaling of the magnification or the scaling of the receptive fields of retina or B1, then you can expect that fovea and periphery can have a similar property of the visual experience. Okay? And that's what you probably experienced in this type of the visual stimuli. On the other hand, just to summarize the last figure, so neurons that respond to the peripheral visual inputs reflect inputs from cones of a wider area than those that respond to the foveal visual inputs. And also, this convergence effect becomes stronger for the neurons at more periphery. Assuming color qualia arise from higher areas, big and uniform surface can evoke stronger color qualia. Okay, so that's the upshot of the last figure. On the other hand, let's think about the another demonstration that I gave. Okay, so here the size of the stimulus is uniform across all these fields, and then in that case, red at the fovea can be perceived as red, like this mechanism. On the other hand, that the peripheral stimulation, like here, projected onto this peripheral patch path, this does not contain the uniform color stimulus, and especially it is interspersed with these gray areas, and it doesn't effectively generate a response from either pink or green or a purple. And then as a result, it doesn't convey the vivid color kind of information. And further worse, these V1 neurons get convergence from other neighboring neurons that also receives an unclear kind of response. And in the end, it doesn't generate very vivid sensation at the color area in the higher area of the brain. Okay. If the same size of object is projected to the retina, then neurons at the fovea have a better spatial resolution, and collecting inconsistent color information at the periphery predicts that more blunt responses of the neurons are responsible for color qualia at the periphery. But there is nothing magical about that. But this is not the same thing as the statement like Dan Dennett made, as we cannot see color at the periphery, or color extends over the edge of the the vision. It's not really, you can't make that kind of claim from just by this type of the argument. Okay, so summary is that of this section is that retinal ganglion cells have a receptive field. That's a very important concept. Once again, I'll highlight that looks like annulus or on center, off center, and send visual inputs to the lateral geniculate nucleus through the spike, through spikes, which is either all or none. And then some are sensitive to onset, and the others are sensitive to the offset of the visual stimuli. And the receptive fields of the retinal ganglion cells scale as eccentricity. And that has an implication for conscious phenomenology in fovea and periphery. So the next part talks about part 2 of this part.